Hey microbiologists, I'm going to demonstrate the next lab, which is looking at microscopic eukaryotes. So normally we work with prokaryotes, right? Cells that don't have membrane-bound organelles, including a nucleus. This lab we're going to be looking at eukaryotes. So even though these guys are microscopic, they're still eukaryotes. They have membrane-bound organelles. They have a membrane-bound nucleus. So you just have to look at maybe like two or three, see what you can do. These are live creatures. This is my brine shrimp. I recommend starting with these today. Actually, these are the vinegar eels because you can see them. They're not technically microscopic, so it might be easier for you to work with. This is called a prep. This prep technique is called a wet mount. We don't have specialized wet mount slides. Ours are just flat. We're going to be using our regular slides. So to keep the water from spreading off the slide and to keep your eukaryotes from swimming either out of your field of view, because a lot of them won't like the light when you get them on your microscope or the heat. So they're going to move out of the field of view, which makes it hard to focus on them. And then the other thing they do is they like to crawl off the slide and into the microscope innards. And we don't want that, so we're going to use this gunk called proto-slow. And I'm just going to put a little circle on here, about the size you usually draw with your Sharpie when you make a field of view for your bacterial smears. And I'm just going to kind of outline a work area. And again, it'll keep my wet mount from spilling off the slide, and it'll keep the eukaryotes from swimming all over the place. Because these guys are live, you kind of have to chase them a little bit. So we're going to use these transfer pipettes. And for these larger species, like the brine shrimp, we might even like to cut off the tip to make it a little bigger. That's one thing we can do if you're having a frustrating time. I'm just going to go ahead and grab one. So I'm going to squeeze this first, and then I'm going to target the shrimp that I want, and then I'm going to suck him up in here. And he's right there. I'm going to get rid of a little bit of that water because I don't want it to flow over the edge of my proto-slow boundaries. I need a good squirt. So I don't really want a lot of the solution on there so that I can really squeeze this hard to force the organism out of that tube. Okay, and then you don't need a cover slip. You really shouldn't be working on any objective that would come into contact with this. If you put a cover slip on this, also it'll limit the movement you can make. I'm messing around with this bubble. I don't want any air bubbles on here because they're just maddening when you get to the scope, so I'm just going to suck those up with the pipette, and then you can return the pipette back. I'll probably have one for each container. And this is the really critical part today. These are all oxygen breathers, so we don't put the cap on tight. We don't even screw the cap on. I'm just going to lay the cap on, kind of balance it with that pipette in there, and just leave it like that all the time. This never gets shut. Otherwise, you're going to kill my babies, and then I kill you. That's fair. Okay. Once you've got your guy on your slide, you can take it over to your microscope. And make sure the stage is all the way lowered. Lowest position. You want to be on the lowest objective. All right, so my stage is in the lowered position. I'm on the 4 power objective, my low power. And then just carefully, because remember this is wet, so if you tip it too much, it's going to spill into the microscope guts. And Again, one of the advantages of starting with the brine shrimp or the vinegar eel is that you can see it quite easily, and I can just get him right in that light. And he's going to wriggle because he's not going to like that light. It's going to get really hot for him. So I'm going to work quickly to see what I can find so I can get him out of that light a bit more quickly. So I'm just pulling the stage up until I see something. Oh, there he goes. These are all swimmers. So unlike your bacteria, it's probably going to be a little easier to find because there's going to be all this movement. And then fine focus may be your best friend in this lab. You're probably not going to be able to get the whole specimen in focus at the same time, so you might have to switch between planes, the right side, the left side, dorsal, ventral sides. The fine focus is going to do that for you. So, okay, I'm going to let you see what I'm seeing as best I can. Okay, so we're not getting a great view, but you can see those are his legs he's swimming with. Let's see if I can get you a view of the head. Come on. Well, the head is down there, but I don't know if I'm going to be able to get that for you. And then you just observe this. You use your data table to draw a sketch. Remember, it doesn't have to be masterful art. Labeling goes a long way. Note color, can note texture, can note things that looks like lots of little legs. Flagella, cilia, anything you see. And again, I'd like you to try at least 3 of them. And if it gets really exciting, I'm going to work on actually feeding these brine shrimp to the hydra. My hydra might be a little too small to be able to eat them, but we'll play around with that tomorrow. And at any point, cut yourself off. About 5 minutes. If you can't get anything into focus, you feel super frustrated, like it's not going anywhere, just call me over. Let me help. I'll figure it out. Okay, see you in the lab, guys.